and thank you for inviting us to join the LEAF network, which is great and a great pleasure. We're looking forward to it. So I'll just share my slides. Whoops. Okay, is that okay? Apart from...
Yeah, that's showing fine, thank you.
Brilliant. Okay, thank you. So first of all, an apology from Dave Chandler and a big thank you to Lauren for stepping in. So Dave and I have been very heavily involved for, I don't know, the last week or so in an EU proposal, which is actually about biopesticides and increasing the availability of biopesticide products. So it's just gone in and we're crossing our fingers now. So my remit is to provide an introduction to Warwick Crop Centre, and I'm sure some of you are already familiar with us and have visited us. We're part of the School of Life Sciences at the University of Warwick, and the school itself is a centre of expertise for all aspects of crop science, but I guess through the activity that we do at the Warwick Crop Centre, a good deal of the focus is on vegetable crops, ornamentals, and some herbs as well. At Warwick, we're probably one of the largest groups of plant scientists in the UK, so there are probably approximately 30 teams working on plants and crops, and that sort of spans the range from fundamental research, so sort of pure science, right through to the activities that we do at Warwick Crop Centre, which we call translational or applied research. We're based on 2 campuses, so a campus called Gibbet Hill, which is quite close to the main Warwick University campus near Coventry, and then also the Wellesbourne campus, which is now called the Innovation Campus, the Stratford Innovation Campus. And most of our applied research is focused at this campus at Wellesbourne. And many of you will be aware that research has been going on at this campus for 75 years. So we actually had some celebrations in July to celebrate our 75th anniversary. And between the 2 campuses, we have a range of skills and facilities to support research on crops. So, as I said, Warwick Crop Centre has an applied focus, and we work quite closely with industry, with farmers and growers, but also with a lot of the businesses that support farmers and growers, including seed companies, agrochemical companies, companies who produce biocontrols. So what I'm going to do is just run through a few highlights, and then Jackie and Lauren will expand on a couple of areas. So just to reflect on the history at this campus, so we're actually older than the university itself, which I think celebrates its 60th anniversary next year. And we started off as the National Vegetable Research Station in 1949, which was a response to food insecurity after the Second World War. And then I guess we've gone through a number of funding bodies and name changes. And then in 2004, we became part of the University of Warwick. And then the Crop Centre was formed in 2010. In terms of facilities, and this is, yes, I mean, a lot of this applies also to the main campus, but here at Wellesbourne, we have high quality labs for all aspects of crop science. We've got controlled environment facilities, so places where we rear insects and where we do controlled experiments on plants and insects and plant pathogens, etc. We've got glass houses and polytunnels, and some of the glass houses have environmental control and containment. And then we have field trials areas, about 40 hectares now of land, which includes some specialist areas. So we've got some specialist pest and pathogen areas, and a low nutrient status field for experiments on the application of nutrients. And then we host, if you like, we host a Met Office weather station, which we have done since 1950-ish. And we also host one of the suction traps that are part of the Rothamsted network. We're also home to the UK Vegetable Gene Bank, and that was founded in 1980 as a result of an Oxfam appeal. It's now funded by DEFRA, and DEFRA also supports the Vegetable Genetic Improvement Network, VEGIN, about which you're going to hear a bit more. So in terms of expertise, then there are a number of areas that we work on. A big focus for all of us now is crop genetic improvement. So the identification of novel traits, and then a genetic analysis of where those traits are lodged genetically. And then we do collaborative breeding research with seed companies. And David was talking about climate change, and at least some of the traits that we're looking for, they include pest and disease resistance, but also our responses of crops in the face of extreme weather events due to climate change. We have a significant amount of activity on plant pests and diseases, so understanding their biology, big emphasis on biological control. I work on pest forecasting, and we also look at integrated approaches to crop protection, so integrated pest and disease management. There's an interest and focus on soil health, so the small organisms that live in the soil, and the larger ones, particularly earthworms. And Jackie is going to talk to you about the earthworm research in a while. We also have an interest in crop physiology, so crop nutrition and development, and then resource and environmental management, so low carbon farming, land use, ecosystem services, nutrient and water use efficiency. And then my colleague, Dave Chandler, who sadly you won't see this afternoon, he also has an interest in honeybees and works on bee and pollinator health. And that's Dave at the bottom left in his bee suit. So in terms of crop improvement, then I've already mentioned the vegetable genetic improvement, and Lauren will expand on it. But basically, we're exploiting genetic diversity in the 4 main vegetable crops, and more recently in other crops as well. And so we're looking for these particular traits, pest and disease resistance, and increased crop resilience. We're also a partner in Origin, which is the oilseed rape equivalent of VEGIN. And again, it's about exploiting genetic diversity to identify sort of sustainability traits. And both of these project programs are funded by DEFRA. Then I thought I'd just talk about the research of a few individuals. So our colleague John Clarkson focuses on soil-borne pathogens, and these are incredibly difficult to manage, and yeah, solutions can be pretty hard to find. So that includes sclerotinia, fusarium on multiple crops, and Pythium species. So for example, cavity spot on carrot. The sort of things that John's team do are plant resistance screening, and then sort of focusing on any traits that are identified, and that's part of the VEGIN project, and then looking at integrated control approaches to plant pathogens, which include fungicides, but also biological control and biofumigation. And then another significant factor is actually being able to identify plant pathogens, so molecular diagnostics and characterization. Then we come on to integrated pest management. So this is mainly focused on control of pest insects. And Dave, I and Andrew Dukes, we work on, say, insecticides and bioinsecticides, both in field and lab trials. Again, within vegging, we work on host plant resistance to pest insects. I've mentioned monitoring and forecasting for a range of pest insects. We look at physical control methods, and to undertake our research, we do maintain quite a number of insect cultures so that we can actually work on them when we want to. We keep many of those in the lab, but we also have field populations. For example, we have a field population of carrot fly. And just to follow on a bit from integrated pest management, then as part of the sort of forecasting and monitoring activity, then I run and have run for a number of years the pest bulletin, which was originally supported by AHDB Horticulture. And this consists of a number of web pages with information, forecasting and monitoring information about the major pests of vegetable crops. And then people can also sign up for a weekly update email just to alert them of anything that's changed. Then move on to Jackie, and Jackie works on earthworm biology and a range of aspects, and she will explain a bit more in a minute what she does. And then Stephen Parnell, who is a modeler basically, and he works on models of how pests and diseases spread through plant populations. Some of his focus is on crops, but also he has quite a lot of focus on things like tree diseases as well. And he also uses models to test strategies to detect and control various pests and diseases. And then finally, in terms of individuals, on to Rob Lillywhite. So basically, Rob works on understanding the environmental impact, the environmental outcomes of different practices, and how the burdens might be mitigated to reduce environmental impact. So he looks at all sorts of aspects of resource use, from energy to fertilizer to plastics, etc. And he uses environmental accounting techniques, things like life cycle assessment, carbon footprinting, to look at hotspots, trade-offs and impacts in different systems. And then on to a very new initiative, which we have called Warwick Agri-Tech. And this is a collaboration between the School of Life Sciences and the Warwick Manufacturing Group. The Warwick Manufacturing Group are part of the university and have a very excellent reputation of particularly working with the car industry. But they're now quite interested in moving into agriculture. And so we've set up this collaboration so that we can explore the opportunities for things like automation and robotics, AI, big data, and all those sort of aspects. And I think Lauren's going to mention one of the projects that we're involved in with them. And then obviously very important to Leith is biodiversity. We try to take great care of the biodiversity on our own campus, and have monitored it historically, and we work quite closely with the local biodiversity organisations, for example the Warwickshire Wildlife Trust, and one thing we're involved in at the moment is partly based on our campus is the South Warwickshire Corn Bunting Project, because we have a population of corn buntings. Personally, I'm also involved in Warwickshire's local nature recovery strategy. At the moment, we have a survey out trying to get everybody in Warwickshire to say what they value about nature. Yeah, we're looking forward to analysing the results. And then in particular, Richard Napier, my colleague Richard Napier and I have been working on a number of other sort of broader aspects particularly to do with fresh produce. One is to do with engaging with sort of national food policy, and with colleagues from other organisations, we developed a strategy paper called Growing British, which was about promoting fresh produce production in the UK. Before the election, we also produced, the same group, a manifesto for growing more British fruits and vegetables. We've been working locally with Stratford District Council on a fresh food strategy for the area. And we're also at the moment developing a fresh produce knowledge exchange hub, which will be a virtual information hub, which hopefully will, for example, mine all the information that AHDB horticulture have generated in the past. And as part of that, we're planning to include a chat box so that you can actually ask it questions. And finally, how can you engage with us? Well, we hold regular webinars on various topics. We have workshops and open days on site, and happy to host other organisations as well. And you can sign up through our website to be alerted about webinars, events, and also to join Vegin as well. So I'll stop there and again say it's great to be joining the Leaf Network. Thank you very much for asking us, and any questions?
Thank you, Rosemary. So I'm just going to be selfish and ask a question myself to start with. So in terms of the work that you've done over the last 75 years, I get the impression that it's gone from more potentially sort of field-based, practical, pest-focused work, and now it's almost as if you're broadening out across the industry with your new partnerships with sort of Warwick Agri-Tech and the policy side. Is that sort of a direction that you see yourselves going in, going more into the policy side and broadening out so that it's not just necessarily crop protection focused work?
Yes, I mean, I think obviously, you know, when National Vegetable Research Station started, our focus was, I guess, scientific research on vegetable crops, and we've broadened our crop areas of interest as well. And I guess with, you know, things like Warwick Agri-Tech, we've also expanded our area of interest. But I think it seems to me in this day and age that you need to be able to, well, engage with the whole, you know, the whole food system and members of the food system, and also engage with policy, because in the end, all of that, I guess, directs the research that we need to do. So yeah, I think we realise we need to be part of it, and involved in yeah, all aspects.
Brilliant, thank you. Right, so if next we go on to Jackie's presentation, if Rosemary, you're okay to stop sharing your screen, and then Jackie, you should hopefully be able to join. So Dr. Jackie Stroud is an assistant professor of soil science at Warwick, and she's going to be telling us a little bit about earthworm ecology and acoustics. So I can see your screen okay now, so hopefully everyone else can. Take it away.
Can you hear me okay?
I can.
Fabulous. All right. Well, thank you very much for the invitation to talk. I thought I'd talk today about our work on soil vibroacoustics. And in England, with our temperate climate, we're very lucky to have fascinating below ground ecosystems with wonderful, amazing things waiting to be discovered. We're the first species with wholesale control on our hectares of land, from the plants we sow and every mouthful of food they provide, to how we cultivate the soil and influence the worms and the charismatic predators that they feed. And the challenge is and continues to be taking meaningful measurements. And today, that's what I want to share with you, the journey that we're making to develop soil vibroacoustic research to make it as useful as it is fascinating. Now, one of the most intriguing soil sounds is really quite mind-blowing. You're probably all familiar with the cuckoo. It's a bird which lays its eggs in the nest of other birds. The hatched chicks then push the host eggs out to be raised with the host chicks. But in the soil, this behaviour is exhibited by maybe 10,000 species of butterflies, beetles and fly larvae, with dastardly behaviours which trick subterranean ants into looking after their young. The best example is a type of butterfly found in Austria called the mountain Alcon blue, and its larvae emit a chemical signature which trick ants into carrying it into their nest. The parasite then lives with the ants for 1 to 2 years before it emerges and is looked after favourably by the worker ants. And when I say favourably, nurse workers can kill and feed their own brood to this parasite when food is scarce, or rescue the parasite in preference to their own brood when disturbed. The trick that this larvae it has, and the pupae has, is mimicking the sounds made by the queen ants, which worker ants respond to. And the parasite, you see, is communicated. It has a high social status, allowing this intimate relationship to develop. And they go from perhaps just 5% body weight to 95%, being fed directly with the regurgitations from worker ants. Now, closer to home in the Polden Hills, there's a similar butterfly species, the large blue, which has been reintroduced after near extinction. Its larvae seem to have the same acoustic noises as that mountain blue butterfly, which enable it to live in the nest of red ants. This larvae is not a cuckoo. It just uses the noises to live alongside in the fringes of the red ant nest and makes forays into the brood chambers to feast on the ant larvae. And these evolutionary couplings, they're not always just one-sided. In Australia, there are ants and butterfly larvae which live together and use acoustic cues to help each other. And the benefit to the butterfly larvae seems to be defense from predators, from the by the army of ants. But it's really quite amazing that invertebrates have these really sophisticated vibration systems, and this influences the evolution of soil-dwelling organisms together. And these evolutionary couplings then produce those delicate blue wings frittering over grass that people tend to value. And in fact, many soil insects intentionally produce vibrations. There are 2 types. There's stridulations, which is where they rub body parts together. And their closest example I can give you is sort of clicking your fingers. And the other type they do is the broadband vibrations, which is drumming the substrate, so like that. And these alarm signals, the sort of drumming, is most well understood. It's a bit like a dog barking at the door when something's nearby. And they can be really quite sophisticated. For example, termites signal the presence of fungal pathogens to reduce the infection risk in their colony. And the vibrations themselves are likely to be felt as a sort of pushing sensation from where the animal resides. And this displacement is what we're able to detect using sensors. Other sophisticated interactions include sort of eavesdropping. So, for example, termites can eavesdrop on ant footsteps and specifically avoid them in soil habitats, whilst entomopathogenic nematodes can locate insect hosts using vibrations. And it's sort of little recognised that these vibrations in soil, so it's not sound, it's substrate-borne vibrations influence social organisation, cooperation within and between groups, predator-prey relationships, with signalling occurring, including to signal the presence of pathogens, mates, predators, calling for help, and indicating where there are food resources. And this occurs in a soil substrate that earthworms, the soil makers, continuously engineer, shaping the propagation of vibroacoustic waves throughout the habitat. And actually, this vibrational communication, rather than sound communication, is the most ancient and taxonomically widespread form of communication, of which chemical with chemical communication, although it's much less well studied. And vibrations are actually emerging as a tool for integrated pest management in open field systems. It's been done a couple of times now, but this novel technology is assumed to be environmentally friendly in comparison to pesticides, for example. And it essentially just means vibrating plants with shakers with the idea to improve plant health and productivity. There are 3 development strands there: reproducing predator vibrations to repel or reduce pests, to reproduce pollinator or predator vibrations so the plant tries to allocate resources to productivity rather than defense, and finally conditioning pest predators to enhance their efficacy against plant pests. The challenge is, of course, you're mechanically shaking plants, which likely transmits vibrations to the soil surface with implications to the soil invertebrates and the pattern of food resources for their vertebrate predators. So this technology will need to be deployed quite carefully. And as part of that, we really do need to understand soil vibrations better. Another reason for sort of exploring this, and one of the most pertinent predator-prey relationships we have in our UK ecosystems, is birds and earthworms, as there seems to be a decline of earthworm-feeding birds, including the herring gull, song thrush, and lapwings. And there's also thought to have been a decline of earthworms in Britain. And for example, for the song thrush in farmlands, it's thought to be the inadequate seasonal food resources caused by rapidly drying summer surface soils, which is influencing their nesting attempts and breeding densities. And on the one hand, people are sort of assuming that perhaps earthworm populations have just plummeted. Another idea is that it's where earthworms are. So, for example, earthworm topsoil biomass can range from 0.5 ton to 3 tons per hectare, but it's likely to be pushed up or pushed around the soil by its predators. But it could also be pulled towards the soil surface linked to soil moisture and location of food resources. And so you can have management pools such as having surface cover of decaying plant remains rather than the bare soil surface, having living roots in the soil, cover cropping, reducing tillage, those sorts of things, which can enhance earthworm populations in that upper layer to enable the availability of earthworms to be sufficient to support the predators that rely upon them. Now, in terms of how soil health and soil quality can fit into this, I think it's quite important. Sorry, has my screen moved at all? No, it hasn't. Okay. Birds. There we go. Here we go. There we go. So so soil health and soil quality. What I like to think about is approaching soil health from a rather practical perspective, which is difficult to do sometimes here. But I found this nice word cloud from farmer meetings in Australia, which they collected over 4 years. And the larger words are showing what were the words that were most frequently expressed. So you can see it's things that probably resonate with us too. It's sort of carbon, water, nutrients, organic matter, structure, worms, and microbes. And 2 further insights were that practitioners really place practices before indicators. And this is for the cotton industry, but I think, which is obviously quite different to our temperate farming systems, but the principles and practices look really quite familiar. Maintaining soil cover, reducing soil disturbance, living roots and plant diversity, using strategies such as cover cropping, stubble, tillage, IPM, strategies that are all quite familiar to us. So thus, to gauge the sort of aliveness or life of soils, it seems that being able to just eavesdrop on their communications and movements with minimum disturbance to explore practices is a really nice fit. How this fits into soil health indicators from more of a scientific perspective is slightly more difficult. This is the most highly cited and impactful soil quality publication in recent years, which focuses on indicators before practices. And I just, it always surprises me, but the authors noted, and I sort of read you the quote, remarkably, however, farmers, land managers, consultants, agricultural advisors and other stakeholders usually play an insignificant role in their development, whereas they turn out to be important end users of the schemes. I think that sort of highlights some of the disconnect that can happen when scientists are developing indicators versus working with people to make it quite useful and useful as a technique. So our approach here is very much to approach it both from the indicator perspective but also quite a practical perspective, and we're very lucky here to have the Warwick Crop Centre I've called the sort of sensor testing arena, which is a great place which we've stocked with earthworms and different soil properties, and we've been able to test different probe designs to accelerate the development of technology and to get it into people's hands sooner. And then secondly, here at Warwick is quite unique. I've got a picture of this really, but we have semi-anechoic facilities in the basement where we're isolated from the usual sort of laboratory noises and the hum of machines, enabling us to take really high quality recordings. And we've been working our way through different earthworm species and different model soil types to create one of the largest earthworm activity libraries in the UK, or probably the world, which with our collaborative partner, a consultancy company, is using in their algorithms towards rolling out a service to farmers, which they're costing at around under £1,000 a year towards helping with the interpretation and putting sensors in the field to explore this. And some of the fun facts I can share with you is like the longest earthworm burrowing behaviour we've recorded, and that's by a constant rasping sound. It's been about 19 seconds, but generally it's a sort of short 3 to 4 second burst of activity. And this information is really helping us to strategize how to measure soil vibrations quite effectively. And the topic's actually been quite a popular student project, and I've been able to work with undergraduate students going and visiting long-term field sites. For example, this project here by Rochelle which we're hoping to publish, we found that just 120-second recordings were enough to show very significant differences between clearly different management practices. So that's, we're using that to sort of build the method and get something really robust going into the field. Now, what I'm going to try and do is share you a recording of this. I'm going to play it through these headphones off my phone. It might be a little quiet, so I'll play it twice. And I'll give it a few seconds after each one in case you've changed your volume settings, so to give you time to turn it down. So I'm going to play it now. And stop. And so what you could hear there was the sound going from grass, where you're having a sort of sound event every second, to a ploughed arable setting where you get a sound event sort of in half the recording, and finally fallow, where there was only one sound event in the 7 seconds. So I'll just play it again in case you want to hear that. Yes. Okay. And so the differences suggest that sound analyses which have been developed for things like coral reefs, which get quieter when they're damaged, are really quite relevant and adaptable for soils. And so we're exploring some of the analytical techniques that they use in different habitats and environments, and see whether those statistical relationships and ways of analysing acoustic data could be something we could apply here and really improve our interpretive power. And just for fun, hopefully that works and you're able to hear it, I have a few mystery sounds I thought I'd play it to you. And I'll give you, don't need to say or which one you think it is, but just think about it. What do you think makes this noise? And I think this one's quite a quiet recording. I shall play it twice. So I'm going to, and it will be about maybe 10 seconds or so. Okay, so that one, if you heard it, was sort of some slight sort of popping noises in the middle as it was sliding along. That was a slug moving through the soil. Mystery sound number 2, so does it have legs or not, is this one. So that one was a larvae, not a beetle moving through the soil. And then finally, because earthworms are quite distinctive and almost everybody guesses an earthworm, this is quite a loud recording, so I might hold this a little further away. Decide whether you think it's burrowing in sand or clay, because there is a small difference actually. So that one was, and I find the split's usually 70-30. People are quite good at guessing. That was an earthworm burrowing in sand. In fact, sand, you do get some quite nice recordings in sand. When you move into silty clays, the sounds that you get are a bit more attenuated and they can be harder to hear. But we are able to capture different size earthworms burrowing in the soil. We've managed to record the sounds coming from all 3 ecological groups. And yes, I say we're sort of capturing some of the acoustic properties, so you can look at the amplitude, the duration, towards creating an interpretive library to help understand and interrogate some of the acoustic data that we're generating. And the thing that I'm really excited about in terms of this technique is that we're detecting animals in their own home with minimal disturbance. And with some new equipment that we've got, this can be potentially done across days, nights and seasons. And land use does have a profound impact on the number of vibroacoustic events and also the complexity of those, which we think is linked to, diversity. And then what you might hear, I hope I've sort of shown here. So before I dig holes, I'd look for worms, and you sort of get a number of worms in a soil pit. But now we can look at some of the characteristics of the noises that are coming out over 24 hours, for example. And what we sort of seem to find is that soils are quieter in the morning compared to the afternoon, which is potentially linked to temperature and maybe insect dynamics. And at night, well, that's something completely undiscovered and least well known. And then we could be able to measure across the seasons, whereas normally sort of when you do soil sampling, you'll just go out at peak times. But seasonal analysis will help us to fill in crucial knowledge gaps. And then there's opportunities potentially to link to bird or pollinator noises, because there are many worm feeding predators, and many pollinators do spend some of their lives in the soil. So it could help us to know what works and doesn't work in terms of management. And it would be great to find ways to work together on this. I'm quite inspired by Switzerland, where they have a scheme for the general public where they can book out soil recording units for about a week at a time. And people take them away and sort of listen and share their knowledge. There must be a way that we can do something like that here. And I suppose I'd like to finish by saying that this is quite a different way of looking at soils. And I look back on my own soil monitoring activities where I was sort of digging holes and breaking up the habitat and looking and examining it, to now where I'm putting my field knowledge to good use. It's sort of putting these soil probes into the soil and generally preventing the animal knowing that you're there. So you're getting much more precious insights into this non-human world. And these evolutionary couplings, an example, the butterflies or the songbirds, are sort of signals that our soil management might need some fine tuning, because it's almost certain that some of our behaviours are having desirable and undesirable influences, which are jeopardising our attempts to do good things. And where developing this soil acoustics could be really useful, as well as it could be quite fascinating from my perspective. So I hope you found that interesting, a whistle-stop tour, and thank you very much for your attention.
Thank you, Jackie. That was really interesting. Sorry, I think there might have been an issue earlier where the chat might have not been working for some people, but hopefully you should be able to put messages in if anyone wants to now. Something which I wanted to ask was, so we work on a project in the UK where we're looking at cover crops and the impacts that they can have on soil biodiversity, and something which the project partner said in that is that it's not necessarily just about the diversity of species of worms, but it's also about the number of them, and then importantly the biomass of the worms. Do you think the acoustics could have the potential to get to the point where it could help look at things like abundance and the size of the worms as well as the type?
Yes, yeah, I imagine so. I think for us it's like looking slightly differently at it. So because you're just getting a sort of snapshot when you do dig a hole, looking at the acoustics as a technique, it's we can get longer term trends. So it's looking at those day and night patterns and where you're getting those seasonal activity of worms and those long periods, which would be much more useful than a sort of snapshot of them. Do you see what I mean? So like if we could get that, I think it's a different way of doing it. In terms of interpreting worms and the types of worms, we're certainly working on our library and interpretive power to try and provide some of that information there. But yes, it's sort of like the nice 24-hour periods and the long-term assessments, which I think is going to be quite a different way of looking at soils, which is where it's really exciting.
Okay. And then we've got another question, which is about if a producer ever needed purely financial motivations for increasing worm numbers, what would you say to them? How could you persuade them to care about soil health? For
purely financial benefits?
Yeah, if they're looking at it from a production perspective rather than an environmental perspective, how can you sort of persuade growers to look at soil health?
I think it's hard to say really. If you need to be financial, surely that would be a policy thing. Policy likes giving people money to do things, so I imagine it would be them. A lot of the worm stuff is driven by people who want it done, and then everyone it becomes like something everyone else wants to do. It's sort of led more by popular enthusiasm. But I suppose the government could pay you to find a certain number of worms or something.
And then we've got one more question, which is about, so you mentioned that there's already some companies which are looking at using acoustics to monitor above ground biodiversity. But my understanding from the companies I've seen is that they're generally focused on good insects as such and biodiversity for biodiversity. Whereas could you potentially use this kind of acoustic technology to actually help identify pest species? So, you know, things like the presence of cabbage stem flea beetle in a field. Would you be able to use acoustic monitors to help with pest identification and monitoring?
Yes. Yes, in America, that's been the development trajectory. So they've been using that abroad to look at root chewing critters, particularly in vineyards. That's where the investment has been, and they've been able to show where there are infestations there, and also in grasslands, looking at those too, because that's where the money is.
Yeah, brilliant. Well, thank you for that. I'm going to pass over to Lauren now, who I think is going to speak to us a little bit about the Vegetable Improvement Network.
You.
I can see you, and then
share my screen.
yeah, that's just open
Hoping
now.
is that now full screen?
Yeah.
Perfect. And hopefully you can all hear me okay. Excellent. Thank you very much. Well, thank you very much for the invitation. I'm going to talk today briefly about two of our kind of projects here at the Crop Centre, Vegin and Warwick Agri-Tech. So Vegin is our Vegetable Genetic Improvement Network project. So this is a DEFRA-funded project, and this is all about this kind of interactive network of researchers and industry leaders working together to promote market delivery of improved vegetable varieties using sustainable production systems. Which is a very wordy way of saying we like to work really closely with industry to deliver improved vegetables. We work primarily on the 4 key UK vegetable crops, so carrot, brassicas, lettuce and onion. Vegin has been going for kind of just over 10 years now. We've just received another round of funding from DEFRA to carry on for another 5 years. And actually in this next 5 years, we're including a few more crops to our roster, which will include leek, celery, coriander, wild rocket and parsnip. So Vegin is kind of divided into 2 key themes, abiotic, which is all the kind of climate-based traits that Rosemary and David were talking about earlier. And this work is carried out by our project partners, Harper Adams University. So they're really thinking about all the kind of drought tolerance, cold tolerance, frost tolerance, anything linked with climate, which is of course so topical at the moment. Here at Warwick, we tend to focus more on the biotic traits. So these are all the pest and disease problems that Rosemary was mentioning earlier. So within Vegin, we're trying to develop genetic resources and tools to accelerate this breeding process to get sustainability traits into vegetables. And we do this kind of working really closely with industry and kind of transferring this key knowledge that we develop within the Vegin project. Where does this kind of diversity come from, though? Well, actually, we work really, really closely with the UK Vegetable Gene Bank. The UK Vegetable Gene Bank are based here at Warwick Crop Centre, and they're all about conserving and using vegetable crop diversity. So they're a gene bank, and they have conserved thousands and thousands of vegetable accessions of all the key crops that I previously mentioned and more. So they have over 14,000 different seed samples, a total of over 500 million seeds. So huge numbers of plants there, and a huge range of varieties as well. So they conserve wild accessions, so kind of ancestral varieties of the crops that we work with that you probably wouldn't even recognise as being related to our modern day crops, right through the breeding process to the kind of elite varieties that growers and breeders are producing every day. This is a huge number of varieties, though, for us to be working with in Vegin, and it's not really feasible for us to be working with over 14,000 different varieties every day. So within Vegin, what we've done is we've kind of curated these diversity sets. So these diversity sets are for each of the different crops that we work with, and they kind of represent that range of diversity that the gene bank holds. So within our diversity sets, we'll have wild type varieties, regional land races, right through to some of the elite varieties. And this will represent that huge genetic variation within each of these species that we're working with. So they'll represent all the different shapes and sizes and colours and foliage characteristics. And we can use all that genetic diversity then to kind of understand the genetics and try and identify genes and traits linked to these key improvement traits. So things like pest and disease resistance and tolerance to climate change. Our diversity sets are typically of around 100 varieties, and that's a really manageable number for us to be screening on a daily basis. And it provides enough accessions to represent all that diversity from, the varieties from around the world that the Gene Bank have collected. So I thought I might go through some of the kind of traits that we're currently working on. This isn't all of them, but just a few for each of the key crops that we're working on. So I'll start off with lettuce. You can see in the central picture there, that's an example of our lettuce diversity set, all the weird and wonderful varieties that we hold in that diversity set. And you can see all the different traits appearing. So some of the key challenges that we're trying to look at the moment, pest and disease wise. So diseases, we have fusarium, this causes root wilt, complete plant necrosis eventually. So that's one of the pathogens that John Clarkson is working on, that Rosemary mentioned earlier. On the pest side of things, we're looking at current lettuce aphid, and Harper Adams at the moment are currently looking at things like drought and flooding tolerance in lettuce, as well as pinking and browning. Actually, one of the new traits that we're starting to look at in connection with the fusarium is actually thinking about gene editing and kind of these more modern genetic techniques, and whether we can identify genes linked to resistance in the gene editing approach there. So really exciting novel work that we're looking at there. So in the brassicas, again, our central picture is some of our diversity set. And here in pathogen sense, we're looking at clubroot. So this is a really nasty soil-borne, not quite pathogen, but it responds like a pathogen, and it causes this really typical clubbing of the roots, causing major yield losses, and it, of course, present in all brassicas, including oilseed rape. It lives for a long time in soil, so really challenging, and cultural control is quite difficult, so kind of resistance really is one of the only options there. And on the pest side, things like cabbage root fly, and diamondback moth. Diamondback moth, of course, also linked with climate becoming more of a problem with our kind of warmer summers. So on the carrot side of things, cavity spot caused by the Uomycete Pythium violi is an ongoing problem. The work with John Clarkson, we've made some really good advances in understanding how this pathogen works. And now we're really starting to hopefully understand some of the resistances behind that. Also started recently looking at willow carrot aphid, which is a major problem for carrot growers, but actually alongside that is carrot red leaf virus and associated viruses, which are transmitted by the aphid. So these pests are not only problematic in themselves, but they can transmit diseases which have similarly or equally challenging problems. And again, Harper Adams here are looking at a drought and flooding tolerance in carrots. So moving on to onions. So onions here, we're looking at bean seed fly. Bean seed flies, I'm sure many of you know, is a real challenge. It's a real generalist and will have a go at almost anything. But our onion growers are indicating this is becoming more and more challenging with reducing chemical control availability. Other insects like onion thrip, and we've also started looking at an onion aphid. Challenging problems again, as I said, kind of lack of chemical control means we really need to be looking for resistance here. And things like onion drought and flooding. So what's the kind of system that we have in vegging? Well, we're kind of all about this kind of phenotyping and genotyping. So we work with these diversity sets, and then we develop specific phenotyping assays for each of the kind of traits that I just mentioned. So these are often kind of challenging systems and not well documented with how to work with them. So the expertise at Warwick Crop Centre is all about identifying and understanding the underlying biology of these systems so we can develop these specific phenotyping assays. And we carry out these phenotyping assays in kind of control environment, glasshouse and field. So we're really exploring the full scope of what these kind of plant pest pathogen interactions are holding. There's the graph here shows a really nice example of the kind of results we get from these phenotyping trials. So we screen our diversity set, and because they are diverse, we typically get a huge range of results. And you can see in this graph here, we get kind of resistance and susceptibility. So plants that have high disease and low disease, which is brilliant for us because then we can start to identify those more resistant plants. And that's when we kind of move on to the more genetic side of the project. So genotyping, using existing data and new technology to try and identify those genes, understand the genes, where they're coming from and what they're doing. And once we can kind of map where they are and understand what they are and what they're doing, it's at that point that we hand over to industry. Vegin is all about pre-breeding. We'll do kind of all the basic biology that kind of breeding companies might struggle with. And at the point we know what the gene is and what it does, we hand that over to our stakeholders, all the breeding companies in the UK that we work with, which they can then incorporate into their elite material. They're the experts in breeding, but we're really good at identifying the genes and what they do. So kind of brief summary of Vegin. As I mentioned, kind of Vegin aims to identify these key improvement traits in these key crops. We utilise and understand the novel genetics held within the diverse accessions of the gene bank that we've curated into these diversity sets. We work really closely with industry, and we use these pre-breeding pipelines to share these novel genetics so they get out into the breeding companies, incorporated into the elite lines, so growers can start producing these improved varieties as soon as possible. So I think that's probably a whistle-stop tour of Vegin, and I'll move on to some of our agri-tech work. So Warwick Agri-Tech is a relatively new venture for the Crop Centre, and it's a really exciting collaboration between Warwick Manufacturing Group and the School of Life Sciences. The kind of main vision of Warwick Agri-Tech is to kind of secure the global food system and reduce human impact, kind of advancing research in automation, robotics, AI and plant science. So it's really combining the expertise of Warwick Crop Centre and Life Sciences, that plant, pest, pathogen, basic understanding of plants, etc., with the expertise of Warwick Manufacturing Group in engineering, robotics and AI and computing. And the aim is really to kind of build this sustainable ecosystem and kind of transfer these capabilities into agriculture. So Warwick Agri-Tech, the aim is to kind of really work very closely with key collaborators and stakeholders, and to kind of merge together this kind of plant science with the high-tech technology. So Agri-Tech kind of offers this mix of all the exciting technologies, really. They're very much plant science driven. So we start with a basic question that a grower stakeholder is struggling with. Is it resistance? Is it pest disease problem? Is it something that's part of the supply chain processing? And we understand the plant science driven question, and then we use the technology to try and answer that, whether that's the kind of automation and robotics, AI, data, all these exciting things that we can work together and work out how we can solve that problem. So I thought I might give an example of one of the projects that we're currently working on in Agri-Tech. And this is the Autonomous Spot Precision Application of Herbicide, or ASPA. So this is a Forestry Commission funded project, and we're collaborating with a local grower that produces tree seedlings. And the aim of this project is to develop an AI robot for precision herbicide application of weeds affecting production of tree seedlings. Many of you are probably thinking, I think this already exists. And to some degree, some parts of this does exist, but actually we've taken a very different approach. So current robot weeders use AI to identify the crop, and then they spray everything that isn't the crop, which is great. But as soon as you change crop or your rotation changes, you need a whole new model. So what we're looking at doing here is developing a model that can identify weeds and target specifically the weeds, leaving the crop alone, which makes it much more transferable across all different growers and all different sectors. So this system is really exciting because the aim of this is that we can reduce herbicide use. Being precision spot application means that we should be able to produce a much lower level of herbicide, much level of lower chemical going out into your fields, and far more precise and more accurate, so you can target plants when they're at exactly the right stage. So this project started by doing the AI model development. Agri-Tech have lots of very exciting robots that can measure all kinds of different things. Here you can see one of the camera-based robotic systems that we have. So surrounding this robot, you can see we've got a small lawn of weeds. They were our selected weeds that we chose for this project. So we started off with meadowgrass, groundsel and cranesbill. And we initially sowed seeds in here. So this robot here has been photographing once an hour for 6 weeks, the development from seed right through to kind of young seedling. And this is really important for kind of teaching the AI model what plants look like, how they grow, how they develop, and how you can tell the difference between different plants. So once they've kind of established this model, this is when we started building the actual robot that will kind of go out and about in the field. So this is the robot you can see here. It's quite large. It goes kind of wheeling to wheeling across a bed. And the arms that you can see hanging down are what aid with the cameras to image the weeds and also for the precision spot herbicide application. And you can just see the subset here, this image of kind of blue and red dots. And that's the kind of thing that the AI model is doing. It's identifying the weeds and identifying where that herbicide needs to be applied. So WMG are amazing at kind of building these AI models and incorporating it into these kind of robotics. But the life science side is kind of essential, really, because we need to understand the biology behind all of this. So whilst WMG are building these brilliant robots that can go out and spray the herbicide, there's some core biology going on as well, looking at dose response experiments. So looking at different herbicides and understanding how much you need to kill plants at different stages of growth. And this is really important because when the robot goes out and about in the field, you need to know how long between applications are required. Can the robot just go around a field once every 2 weeks, or is it once every month? And what rate of herbicide is required? Alongside this, we've also been doing germination assays, so understanding how quickly these weeds are germinating and at what temperatures. So that helps us understand kind of how early in spring does this robot need to be going out into the field and start spraying. So you really start to build into the full model so a grower can understand the whole system throughout their year. So hopefully that's kind of, we're still kind of going with this project. We're at a very exciting stage where we're just about to kind of deploy into polytunnel setting, where we'll be utilizing the robot with the specific doses that we've identified in our glasshouse trials. And hopefully that gives you a really nice kind of idea of the collaboration between life sciences and WMG. And actually this kind of Agri-Tech project is so exciting because it's that core biology understanding alongside the engineering that can be deployed to help industry. And that was a very quick whistle-stop tour of the two projects. But if you have any questions, please do drop them in the chat.
Thank you, Lauren. We've got a couple that have been sent through. So the first one is, one of the challenges that's frequently raised in the context of sustainable farming is a lack of genetic diversity in the crops that are grown globally within commercial agriculture. Is this an issue which yourselves are concerned with, and how do you think it could be combated?
Yeah, that's a really interesting question. So kind of when we're breeding, it's really important to kind of understand the genetics that we're looking for. So you'll often hear people are producing new varieties each year that have resistances to key pests or pathogens that are being overcome really quickly. So it's really important that when we identify a gene that's linked to resistance, that actually we kind of have the same approach to that as other genetic diversity, and it does need to be preserved. So it's really important that when we get kind of new resistance deployed, that ideally we deploy that as part of a kind of integrated management system. So you're not just relying on the resistance, you're relying on a whole fleet of kind of solutions to the problem. So you can preserve that resistance for as long as possible. It's really not great if a breeder is breeding for kind of 10+ years to get resistance, and then that's broken in 1 year. So a little bit like Rosemary was describing earlier, it's all about these integrated management systems to preserve all of our techniques for as long as possible, to enable sustainable growth for as long as possible.
Thank you. And then I've got another one about, are there opportunities to use the work within this project to optimize plant development for human nutrition, as well as for plant development and disease resistance? Is this something which you are currently or potentially in future going to be looking at?
Yes, so nutrition is a really important one, and we look at that from two perspectives, kind of nutrition for the plant itself in growing happier, healthier plants, but of course also nutrition for us. Our kind of conversations with key breeders and stakeholders is that nutrition is perhaps not their absolute top priority at the moment. It's growing healthy crops with high yields. It is something that we think about and we talk about regularly. If it's something you are interested in, please do get in touch, because it is a trait that we can look into doing. So yes.
Thank you. And then just to finish on the Warwick Agri-Tech side, what do you yourselves see as the sort of barriers at the moment stopping agri-tech from becoming more common? Is it the lack of government support? Is it the cost to the growers? Or is it just that there hasn't actually been enough research and experimentation done?
I think perhaps what's most exciting about our kind of agri-tech project is the real underlying plant biology alongside the engineering. So that real combination. I think perhaps in the past we've seen a reliance on technology without understanding kind of how plants and pests and pathogens work. So I think there's real scope to expand. I'm perhaps not the best person to ask. I think Dave would be able to give you a much better answer to that one.
We'll have to do a follow-up once Dave is done with all of his EU proposals.
Yes.
Brilliant. Well, thank you everyone for your contributions. It's been really interesting to hear about all of the diversity of research, which is really does quite range from one thing to another. We're really looking forward to working with yourselves going forward, and for any LEAF members that are in the group in the webinar today, we're looking forward to be able to take all of this knowledge which you guys are generating and bring it back out to our membership base. So do keep us updated, especially with the new chat bot that you're looking at to help growers. I think that could be a really exciting opportunity for the fresh produce sector. So I don't know if David wants to say a few closing words, and then we will let everyone get home.
I just thank you, Meg. A couple of things I'd like to say. Number one, thank you. What an extraordinarily interesting hour. Those presentations were absolutely fascinating. And I think, you know, the breadth of the type of work that you're doing, and just so incredibly refreshing to see it so applied as well and so practical. It does sometimes I do slightly despair that we spend so much time looking at things which seem so far away from actually being able to have an application on a farm to drive change, and this is so real world. So really absolutely fascinating. Thank you so much indeed for sharing with us. We have got the LEAF conference coming up on the 6th of February next year. I will be knocking on your doors to ask for people to come and potentially help us out in terms of making the case. So Rosemary, do expect an email. And finally, I'd like to say thank you to Meg as well for bringing this together. I mean, I'm absolutely delighted that you've made this happen. I know there are changes afoot in terms of yourself, and bringing all of this together for us with the team at Warwick in the time frame that you've done is just tremendous. I'm delighted. Thank you from all of us at LEAF as well, to Meg and to Warwick. And we look forward very much to working with you all in the future.